Dr. Juana Diaz from Pompeu with RNA-based therapy therapeutical platform. So thank you very much to everybody, and we wanted to be brief because we would like you, the audience, to make as many questions as possible. So without anything else, I would like to start with Laia, and I'm sorry I call you Laia because we know each other quite well. So please, the floor is yours. Well, this is not a mistake. It is just because so you can remember all the slides, so you can vote properly later.
Okay. Okay. So thank you for the organizing these talks. As you know, I am leading this project together with Professor Antonio Torres from the hospital clinic. We are settled in IDIBAPS here in Barcelona, and we have a big challenge here. In which we are identifying SARS-CoV-2 epitopes in order to be able to produce monoclonal antibodies for treatment. We have used overlapping predictions of 3 different methods in order to find those epitopes with higher accessibility for the antibodies. In parallel, we have collected as well serum samples from post-COVID asymptomatic, mild, and severe patients. And we are, by a Luminex assay, immunoassay, we are identifying which are the epitopes that have a highest binding to the antibodies that in the reality our population with COVID have produced. With this, we will isolate these antibodies through high-performance liquid chromatography, and we also will describe the X-ray quaternary antibody structure and the sequencing by tandem spectrometry. We can move to the next one. Okay, as you know, in spite of the high similarity with SARS-CoV, the SARS-CoV-2 has completely created a pandemic, and because of the high transmissibility and infectivity. In the top right of the slide, I put some examples of these epitopes that we have generated, so you can see how are they located in the spike protein, and see that 7 of them, of these examples, are allocated in the region binding domain, which is the domain that reacts with the receptor. Going into a deeper detail, what we have done is we have created we have coupled our peptides with a polymer PEG12 and biotin complex, and we have coupled this with magnetic beads plus avidin in order to perform our Luminex-based immunoassay. And this assay is what is telling us which are the epitopes that are producing highest binding to the antibodies of our post-COVID population. See below, for instance, how the peptide epitope 2 has produced much higher luminescence in much higher number of patients compared to the epitope 3. And this is what we are doing. We are identifying potentially 5 or 6 best epitopes candidates in order to produce these monoclonal antibodies. We can move to the next slide. And on this furious race against COVID infections, I would like to highlight that, of course, we are working in a completely accelerated timeline. You know that in a typical timeline for a vaccine is 16 years or 15 years, and the goal of the entire scientific community is to have available new treatments and new vaccines in short, during the 2001 or early 2002. So this is a completely difficult challenge. But what we have up to now is what we have learned from, for instance, from current therapies, and regarding antibodies, we have the use of convalescent plasma. In the United States, more than 20,000 patients have been treated with convalescent plasma of post-COVID patients successfully with very few side effects, and We know now that it's very important that these antibodies, of course, are against the target, that we administer enough amount of antibody, and that this administration has to be early, optimally in the first 3 days of hospital admission. So these are good news for us, and we hope that this cocktail of monoclonal antibodies can have prophylactic and also therapeutic benefits for instance, healthcare professionals or high-risk and severe patients. There are currently 2 phase 3 trials that I would like to highlight that you probably know. One with a combination of 2 monoclonal antibodies, and then we have another one with a single monoclonal antibody of Lilly. And just to finish, we need to bear in mind that these monoclonal antibodies have to be prepared for to avoid the mutational escape that is obvious that is occurring in this coronavirus. Let me mention that, of course, we are a big consortia. We are collaborating with IS Global, with the SIG, with Barcelona Supercomputing Center, and with IRB, and I am very happy to have all these people in the team. Thank you very much.
Well, thank you very much, Laia, for presenting this very interesting project. And now we go to Giuliana Magri from IMIM. Thank you very much. Dr. Magri, you are muted now. Okay.
Yeah. Okay. So good afternoon to everyone. So today I'm gonna briefly introduce our project that has been recently granted by the Department of Health of Generalitat de Catalonia. So the work team for this project included 2 very talented researchers at the B-Cell Biology Lab, Dr. Leire de Campos and Dr. Sonia Tejedor, a clinician at Parc Salut Mar, Dr. Judith Villar, and 2 data scientists at GRIP, Dr. Laura Furlong and Dr. Janet Pinheiro. So the idea behind this project is to use the patient's own immune response, humoral response, as a template to develop and engineer in vitro neutralizing antibody to SARS-CoV-2 that can be used as a therapeutic drugs to block and prevent the infection. So the first goal of this project is then the characterization of SARS-CoV-2 specific B cells and antibody dynamics during natural infection in moderate and severe COVID-19 patients, as well as in convalescent individual. And then we will use the information generated from this initial analysis to establish the best pipeline to generate in vitro safe and effective human recombinant virus-specific neutralizing monoclonal antibody from the PBNC of convalescent individuals. Next slide, please. So we initially perform, analyze and profile SARS-CoV-2 specific antibody response in a serum sample from a cohort of patients with moderate and severe COVID-19 disease. And we do that by performing isotyping and in-house developed isotyping and subtyping ELISA using the spike, the receptor binding domain of the spike protein, as well as the nucleocapsid protein as a substrate. So as it's shown in this heat map, based on the SARS-CoV-2 specific antibody profile, we were actually able to cluster patients into different groups. Then we apply a machine learning method to define the minimal set of immune features that distinguish patients with moderate from severe COVID-19 disease. And we found that indeed patients with severe disease have a significantly higher RBD-specific IgG1 and IgG3 antibody, whereas patients whereas a dominant RBD-specific IgG2 response associated with a more favorable clinical outcome. Next, please. So in parallel, we are also analyzing in peripheral blood lymphocytes by conventional and spectral flow cytometry, peripheral lymphocytes of patients in the acute phase of infection, as well as 2 months upon symptoms onset. And the preliminary results from this initial analysis suggest that indeed acute COVID-19 infection is associated with transitory changes, mostly with transitory changes in circulating lymphocytes, including expansion of activated CD4 and CD8 T cells, as well as a huge expansion of plasmablasts. Also, we observe an expansion of extrafollicular memory switch B cells that tend to persist also during the convalescence phase of infection. And this can be relevant for the development of neutralizing antibody. Please, next slide. So to develop human recombinant SARS-CoV-2 specific neutralizing monoclonal antibody, we have set up a protocol to detect RBD specific B cells in the blood of convalescent individuals. So as you know, the receptor binding domain is the part of the spike protein that is responsible of the binding to the ACE2 receptor on target cells. So often antibodies that recognize the receptor binding domain are actually able to block the entry of the virus and are neutralizing. So as you can see in this slide, we can able to detect RBD specific B cells in convalescent individual. Those cells actually persist up to 6 months after infection. And also we found that mainly consist of memory IgG positive B cells. Next, please. So we use the same cohort to develop a first batch of virus-specific monoclonal antibody. So we stain PBNC from this convalescent individual with a mix of antibodies that are specific for B cell marker, as well as with our fluorescence labeled RBD probe. Then we single cell sorted RBD specific B cells into 96 well plate. Then we got the cDNA from each one of these cells, and by PCR we have amplified the variable region of the immunoglobulin heavy and light chain. So this is unique for each one of the cells and is the part of the antibody that is responsible of the binding to the antigen. So then we are sequencing right now these amplicons, and then with selected amplicon will be vector ligated into IgG encoding plasmid, and then co-transfected into 293 mammalian cell line. So each one of these cell culture will produce in their supernatant a monoclonal antibody that can be tested for reactivity and also for neutralization activity by in vitro and in vivo assay. So next, please. So we think that our initial characterization actually will lead to the identification of the antibody property that best correlate with disease recovery and protection, as opposed to disease progression, including the induction of potential induction of inflammation or antibody-dependent enhancement of infection. And this will guide us the definition of a very straightforward strategy for the development of virus-specific monoclonal antibody, including the selection of the best Ig responder, the selection of the best virus-specific B cells, and when we have a candidate, also the antibody optimization to maximize immune protection and safety. Please, next. So right now, the generation of a virus-specific monoclonal antibody is indeed recognized as one of the most promising approach to fight COVID-19, and we think that may constitute an important therapeutic bridge until a vaccine becomes available. Also, once a vaccine is available, neutralizing antibody can still be useful as may provide protection in immune-compromised individuals. And despite the high cost, if we are able to generate a neutralizing antibody with outstanding neutralization activity, that can be used at lower dose, and this will reduce the economic burden on the health system. So, thank you very much.
Thank you very much for a very clear presentation. And let's go to Dr. Rafael Mañez from Belviche, please.
Well, good afternoon, and thank you for the invitation. Well, we'll be presenting certainly a new approach, totally different approach to generating protective immunity against SARS-CoV-2, that is basically the removal of non-neutralizing antibodies. Next slide, please. We know that antibodies are one of the main mediators of the immune response against all the infections, and certainly the SARS-CoV-2 is not really the exception. Once we have the infection, we generate different antibodies against the virus. As we are showing here, we are producing antibodies against, for instance, the spike protein, that is the one that interacts with ACE2 receptor to infect the new, well, affected the cells. However, there is a well-known phenomenon that, particularly in the case of viruses, when the level of antibodies are low and not all the receptors that cover, may happen this phenomenon known as antibody-dependent enhancement of infection, in which what occurs is basically that the antibodies really, cannot neutralize the virus, and what occurs is that the antibodies, there is a reactivity between the Fc fragment of the antibodies with the Fc receptors in macrophages and monocytes, and really facilitate the infectivity. This has been very found in all the viruses, particularly in all the coronavirus infection, and also in all some flavivirus, like particularly dengue disease. Next slide, please. Along with the specific antibodies, we are also antibodies known as natural antibodies, are polyreactive antibodies that also play a role in the identification and binding to viruses. And some of these antibodies may also be really act lack of neutralizing capacity, and may perhaps act as also facilitator of infection. There have been already several studies, the most recent one, this one published in the New England Journal of Medicine last week, showing that there is a clear relationship between the blood group type and the development of severe diseases in the coronavirus infection, and particularly that blood group A, in other words, people having anti-blood group B antibodies, really have a higher incidence of severe disease. Next figure, please. Our group had been working for several years already in a strategy to remove different antibodies that are really harmful antibodies or deleterious antibodies to the individuals. We have developed a platform constituted by polymers to which we can attach different antigens, the specific antigen of the antibody that we want to remove, and that so far are mainly sugars. And these glycopolymers can be injected intravenously or subcutaneously. They initially remove the antibodies, that circulated antibodies. The immune complexes that we generate are metabolized, in the liver, are removed through the kidney. And also, they also bind in the lymph node and spleen new the B cells producing these antibodies. What means that we only are not removing circulated antibodies, we are blocking the production of new antibodies, in this case, those specific antibodies that we are interested. Next slide, please. Our approach that we've been working is we are trying to identify those non-neutralizing antibodies binding the different structure of the virus, and particularly those anti-carbohydrate antibodies that bind the shield that all these viruses, and particularly coronavirus, the glycan shield that this coronavirus that they have. We are using the glycan array technology that allows for identifications. We have a panel with 750 different carbohydrate sugars, and this allows us, using inhibitory assays, to identify those antibodies that bind the different viral structures and which one they lack of neutralizing activity. Once we have identified these antibodies, we can use our polymer platform to generate the glycopolymers, and then next figure, we can, what we are using is try to see whether the specific removal of some of these antibodies, as one suggested so far, the anti-blood group B antibodies, could really enhance the neutralization capacity of human serum against the SARS-CoV-2 virus. The next slide. We think that the summarized is that, well, we are proposing really a game changer, a way to treat infections. It's probably a change of paradigm. Will be the first time that we are proposing to generate protective immunity against infection by removing existing antibodies in a state of producing no ones. Our treatment could be both preventive and therapeutic, because certainly we can remove those antibodies generated during the infection that may also act as non-neutralized antibodies, and of course, those natural antibodies that really act and facilitate the infectivity. And one of the clear advantages, we can remove, these are all the antibodies in 1 hour after the injection, what means then we are able to generate an immediate immunity against the SARS-CoV-2. And this is certainly one of the advantages and why we can be used, as said, can be used either as preventive or therapeutic treatment. It's safe. We have non-human primates treated for prolonged periods with the removal of specific antibodies with no side effect, and we already have well-defined dosage and also way of administration that, as I said before, could be either the subcutaneous or intravenous administration. Thank you very much for your attention.
Well, thank you very much, Dr. Mañez. So let's go to the next project. That would be Dr. Nuria Montserrat from IBEC.
Okay, good afternoon to everybody, and thanks for the invitation to present part of our work. I hope to convince you about the utility of bioengineering and how to grow tissues in the laboratory to start to understand and target SARS coronavirus 2 infection. Something that we think it's wonderful, next please, from the nature of pluripotent stem cells is that these cells that can be found naturally in human embryos, and we can also derive this kind of cells in the laboratory, is that these have the capability to give rise to all the cell types in our body because of their inherent properties. This knowledge has been very useful in the laboratory to develop in the next animation organoids. These organoids are three-dimensional like tissue structures that are composed of multiple cell types. And the magic of these systems is that the cells can be encountered in a very similar fashion as we discover the cells in the organ that we like to mimic in the Petri dish. Importantly, different laboratories, including our laboratory, we have been also showing that the cells that are in these structures are also organized in a spatial fashion very similar to the one that we find in the organ. And in the next animation come the major questions. Can we profit from these mini organ-like tissue cultures to start to understand how SARS coronavirus 2 infects human cells? And also, can we think in exploiting this technology to further understand how to model COVID-19 disease? So these questions were important for us already in February when it was discovered, especially in China and different countries all over the world, on the sequence, genomic sequence of SARS coronavirus 2 and the similarity of SARS coronavirus 2 with SARS coronavirus 1 that we know and we knew already that uses mainly ACE2 receptor for entering into the cells. So our hypothesis was that we could block the entrance of the virus by using a compound that was already tested in a first one clinical trial in healthy population. This compound is a soluble recombinant protein for ACE2, mimicking specifically the chemistry of this molecule. So then we hypothesized to use these microorgans, kidney organoids, and also vascular organoids to infect the organoids with our collaborators in Karolinska Institutet in Sweden, and to check for the possibility to block viral entrance and replication. And this is what we show very early in April, that we could infect these cells majorly because human organoids contain proximal tubular cells, very similar cells of the ones that we find in the kidney. And once we infect the organoids, we could treat these organoids with this clinical tested compound that nowadays is in a second phase clinical trial in 6 different countries. So if you put the next animation, it's more to tell you that we did publish this collaboration very early in April, beginning of May, and now just very recently, we published on the efficacy of the compound in patients in the journal The Lancet Respiratory Medicine. And nowadays, what we are doing with CRISPR-Cas9 is to delete or completely abrogate the expression of other putative receptors for SARS coronavirus 2. Not only ACE2 has been involved in the capture of the virus, also other molecules, as CD147, for example, and other lectins. So with CRISPR-Cas9, we are having the possibility to totally delete or abrogate the expression of these molecules. And in the video that is there just in the next animation is for showing to you how an organoid looks like infected by the virus in yellow. And we are now testing on the effect of different compounds in the background of engineered organoids, also using genetic tools. In the final slide, in the third slide, I would just like to highlight on the possibilities, the theoretical possibilities of using kidney organoids. We can start to study SARS coronavirus 2 host interaction, for example. We can explore for systemic conditions. We are also having other projects in collaboration with our partners at Karolinska Institute, also in the University of Heidelberg and in Vienna, in where we are testing on other comorbidity-related issues like diabetes or hyperlipidemia, on how it impacts on the infection per se on these kidney organoids. Using bioengineering, we have been really taking advantage of different approaches for growing the organoids, and in the last animation is just for showing to you that we can exploit kidney organoids together with CRISPR-Cas9. So I think we have, okay. So in the last part of the slide, you can go all the way because otherwise it's very difficult for me to speak. Okay, thank you very much. So it's more for telling you on not only on the scientific impact of this kind of application for studying COVID-19 disease, but also on the social relevance of this type of project. It's clear that from the scientific side, we are working in an emerging field. We are combining different tools that are innovative with regards of the use of organoids and CRISPR-Cas9 engineering. But for us, what is super important is on the social relevance of the project. So we are working together with different institutions. We are working always keeping in mind the European community, research community indications in terms of gender balance, including in our research and also in our working teams. And for the industrial side, on the industrial impact, all of the different issues that we are playing with in the project have also a long-run framework. So I would like to conclude with saying a big thanks to you and hoping that I convince you on the possibility to make use of mini organs to study SARS coronavirus 2 infection. Thank you very much.
So thank you very much to all the panelists. I am sure that we will find amazing science and game changers in the other panels, but my bet would be that we can find both of them here. Thank you very much for the clarity, but as well for the very innovative approach that you are using in your different projects. I am being said that, well, of course, first of all, that these votings, you can do them now during the questions and answers of this panel. As you heard before, Jordi is very much interested in this polling, so please go ahead with the voting. But as well, now is the moment for questions and answers, so if you have any, please do them in the chat. In the meanwhile, I see that here in the panelist chat, there is one question from one panelist to another, and maybe we can share this one. Dr. Diaz was asking something to Nuria Montserrat in relation to the organoids. Maybe you can share that with the rest of the audience.
Oh yeah, I would like to ask you, related to the organoids, it was not clear for me if these organoids mimics some kind of organ specifically. I mean, they could mimic lung, they could mimic, or it's just general. Could you please explain to us?
Yes, the technology of organoids, it's been for the last 5 years, I think, one of the major booms in the field of stem cell or cell biology. We can derive organoids from adult stem cells or from pluripotent stem cells. In the laboratory, we are focused on developing organoids from human pluripotent stem cells, the ones that can be derived into any cell type of our body. But other groups in the world are also deriving lung organoids, and this is not new. The world has been advancing quite a lot using mouse embryonic stem cells, and you can also find work on lung organoids from human pluripotent stem cells. And now, because of this research that we are doing with our collaborator in Vienna, we are also running organoids for developing lungs. But we need to consider always that these are quite embryonic structures. The field itself is still under development. So we are very autocritical in that regard. The organoids can be very helpful, for example, in the examples that we have been putting for this recent work that we did publish together, with regards with the specific cell types that we encounter. Tubular cells within the organoids look much more like the adult cells that we find in the human kidney. But if we are looking for cells that are going to be producing hormones, for example, inside of these mini kidneys, we are still far to be there. So the field itself is evolving, so we are still working on that. But for some specific questions, like the ones that we have been touching here in this meeting for the SARS coronavirus 2, as H2 is highly expressed in proximal tubular cells in the native kidney and also in the proximal tubular cells in the kidney organoid, we are working side by side with both human from patients, cells from patients and from organoids, and we see that the correlation is great. But for other organoids like brain organoids and other organoids that people are developing, the technology itself is still quite new, but so far so good. I mean, we are not the only ones, fortunately. We have a lot of competitors, but now all of us, people that were competing in January, we are all working together. So I think it's also nice to see that we are putting forces together to put all the organoids for the same question, right? So how to stop?
That's actually, it's great system. I really am very interested, because really you were growing in a 3D structure and we are used to this 2D, and also, I mean, the typical work is made with transformed cells, no? I mean, all the cell lines are cancer cells, so we are working in cancer cells. So this it's great to have this system. I'm sure it will be a long way, a lot of things very interesting to be found there. Thank you.
Thanks to you. Thank you.
Okay, I was seeing Jordi smiling because we are seeing networking in action now and synergies, possible synergies that might arouse from here. So that's good to see. It seems that we don't have questions from the audience. What we would like you is to keep on voting for your preferred beautiful science and game changer projects. I would like to, I have a question maybe to, and it is not so scientifically, but maybe more about innovation to Laia and Dr. Fernandez Barata and Dr. Magri in relation to the scalability of the production of monoclonal antibodies and the pricing as well in the long run. What could you tell us about that? And how far you might feel from that stages as well.
Okay, I can answer something about this. Antibodies are very expensive, so this is a very good question. Are very expensive, but I don't know if in the problem that we have now, the high production could, of course, decrease the price. This is the question that we need to have in mind. No, I'm sure it's different. It's a different problem. There is a pandemic, so I imagine that the price would decrease. Yes. So this is my answer.
Juliana.
Yes, yes, for sure is an expensive drugs. That's clear. I think, and this is something that I try to emphasize in the presentation, that we can do a monoclonal antibody that can work at nanomolar concentration. So that means that you can really use a little. So it's not like the 8 grams that have been used for Trump. So this is the idea. So I think that in along the time we can get much better tools that can be used a much lower concentration. Also, there is somehow, and this is something that is related also with the project that I present, there is the idea that maybe we can also think on therapeutic drugs like monoclonal antibodies that are not going to be administered intravenously, but can be also used as a kind of microbicide that can be used orally or in the upper respiratory tract that can block the enter of the virus. So maybe then you don't need to use as much as you can use. You have to use for if you are administered them intravenously. So I think that there are already possibility and strategy that make them a feasible option, let's see.
Thank you. Well, thank you. And maybe another question to Dr. Mañez, if possible. I understand that you have been using this technique for other purposes in the past. What differences have you seen with SARS-CoV-2 since you have started working with it? That would be the question. And as well, if there is there any actual drug that works with that very innovative techniques? I'm sorry, my ignorance for that.
Well, this is certainly a new approach. We have a very drug that is in a very advanced stage of development. And this drug basically removes also the glycopolymer that we have, that we are developing to prevent the gram-negative infections in patients, ICU patients. That drug is ready to enter in phase 1 clinical trial next year. And we are already making all the escalation. We have a Caixa Impulse project, and we are working in that. And this drug basically removes also anti-B antibodies. Therefore, is one of the ones that we are tested right now, because as I say, this one is probably will be ready to next year. But anyway, we are also looking for other antibodies, in the sense that we are basically the advantage that we have in our structure immune disease. We have this platform now with different polymers to which we can attach different antigens, mainly sugar antigens. And anyway, if we really show that what antibodies are really the antibodies responsible, the non-neutralizing antibodies that act as facilitator of the infection. In the glycan array study, we already finished. The study is certainly under analysis by bioinformatics now, because we did over 100 patients admitted in the ICU with severe COVID disease. We are doing the glycan array technology. We already with 700 different antigens, and we are now trying to see if we identify other. Certainly for us, we can probably in 3 months may have new glycopolymers that can also be tested. And as I said, and since we are ready to begin in human clinical trials, I think the safety, of course, here we have large numbers of primates treated. And one of the advantages, and perhaps linking with your previous question, is that the technology is a very cheap technology, because it's a synthetic molecule. And even that the synthesis of sugars are not really easy, but compared, for instance, with all the other technology, our technology, for instance, will be even cheaper than the use of any generic antibiotic, for instance, used to treat infections. Therefore, it's a very cheap technology, what means we are able to show that we can generate immunity, and really it works in the prevention or in the treatment of infections, bacterial, and of course here in this case, the one we are interested in, the SARS-CoV-2, our approach will be really relative. It's a very cost-effective approach.
Well, thank you very much. I understand as well one of the answers before nailed. Well, it is different of producing monoclonal antibodies for, I understand, for different pathologies, but in an emergency situation, I understand that they could be cheaper produced. But I understand that. Yeah, thank you very much. And last question to Juana Diaz. There's a question from the audience, and it is about. And I'm sorry, I received it. The question is about the platform. Can you comment if the nanoparticles that you use to deliver the plasmids, are they FDA or EMA approved already?
Yeah, they are nanoparticles to deliver RNA, okay? Because what we deliver is RNA based. Yes, they are. It's actually a commercial one. This is not a I mean, we chose a commercial one to be, to make things faster. So it's named JetPay. So it's no problem at all to say it.
Okay. This was a question from Annabelle Sanz. So, well, thank you again. I think that we have to keep on moving. So thank you again for your very interesting and clear presentations. And Jordi, over to you.
Yes, thank you very much, Ignacio. Thank you to all the panelists. I think that voting is ongoing. Please scroll down the screen to see all the different projects and be able to vote. I think we'll have difficulties at the end of the day in choosing both recognitions of beautiful science and game changers. I think we have seen, I would vote for the for the 10 projects that we have seen already for both categories. So it's going to be a very difficult selection. I can tell you that we are more than 200 people connected. So we are 200 people following this seminar. So thank you for your patience. A special thanks to the Houston Control Center in Girona. I mean, half of the team is there and the TV producer is there. So as you can see, we are finding a second job as TV producers and anchor people in TV programs like this one. But we do enjoy very much. So, well, now it's time for some commercials, for a bio break, some ads. Let's finish voting the Beautiful Science Game Changer project. Let's also see how the voting for the first vaccine to be so chimpadenovirus platform is doing good, although the potential, I mean, all the hiccups in terms of safety during the clinical trials. Please, Moderna team, BioNTech team, Chinese team, Novavax team, or go ahead. I mean, no, it's not lost. It is not lost yet, the match. And so, bio break. Let's reconvene. Please, I mean, the 200 of us. I know your names. Don't leave the program. Don't go to the next Teams or the next Zoom. Tell them that you're late, that you're attending a much more interesting seminar this afternoon, this evening. And let's reconvene in 55. I mean, at 4:55, like in 5 minutes, right?
Okay.
Bio break, 5 minutes. I want to see everyone here now immediately after the bio break. Thank you.